Welcome, dear students. Today we are going to talk about an interesting topic on super domestication of plants. As human societies have evolved, so have the plants in the human environment. The transition from gathering wild plants to cultivation involved increasing interaction between humans and the plants they used. The subsequent genetic changes in these plants resulting in domestication of some of these cultivated species reflect the genius of early farmers, who were the first plant breeders. The present generation of plant breeders have tools available that enable them to be plant engineers. Cultivation of domestic plants, when mankind propagates plants and harvests them, to have played significant roles in many of the advances that pure and applied botanical sciences have made in the last few centuries. The earliest farmers recognized useful genetic variation that could be chosen from the wild, planted, harvested and reselected in order to gradually develop improved populations with a range of desirable traits. The domesticated forms bore only limited resemblance to their wild ancestors due to the selection of domestication genes. Early in the 18th century, the first conscious hybridization of plants occurred using the cultivated ornamental species Dianthus. Knowledge of plant hybridization paved the way for the use by plant breeders of intra and interspecific hybridization in crop improvement. Mendel's discovery of the laws of genetics, based most famously on his experiments with the domesticated garden pea, led to the improved understanding of the variation in domesticated and wild species. Subsequently, sophisticated crop breeding programs were developed that enabled more efficient introduction of desirable traits from one cultivar to another. Knowledge of the evolutionary relationships between crops and their wild progenitors has facilitated more efficient exploitation of the genetic resources represented by the wild relatives of domesticated species. Currently, domesticated crops such as rice, maize, and tomato are major targets of studies in molecular genetics. Research on domestication related traits is leading to a better understanding of how genetic control of phenotypic differences is affected. For example, in determining the extent to which similar genes are involved in producing similar phenotypes in distantly related species and working out how to transfer desirable genes between species that cannot be hybridized sexually, and then how to control the expression of the genes once they are transferred. Hybridization and selection have both been involved in the origin of crops and the process of domestication since early times. Bread wheat, which is a hexaploid, that is 2n is equal to 6x is equal to 42 chromosomes, arose through fortuitous hybridization between tetraploid wheat, that is Triticum turgidum, subspecies Dicoccum 2n is equal to 4x is equal to 28, and diploid goat grass Agelopus thauschii, that is 2n is equal to 2x is equal to 14, a weed of early wheat fields. New groups of bananas and plantains developed when diploid domesticated bananas with genome AA spread into the range of wild Musa bulbiciana, that is genome BB, producing the AAB and ABB triploids. Modern strawberries, that is Fragaria ananasa, are a consequence of hybridization between North American Fragaria virgiana, and South American Fragaria chilenensis, when those hitherto geographically isolated species were cultivated in close proximity in European gardens. Selection, both intentionally by humans, that is conscious selection, and as a result of environmental factors, that is natural or automatic selection, has in most crops established the traits associated with the domestication syndrome. Analysis of the genetic, genomic, and molecular basis of the traits selected by early farmers that constitute the domestication syndrome in crops, such as loss of seed shattering and increased organ size, has been a major focus of much recent research. Profound insights into traits associated with crop domestication have resulted from the technologies and discoveries that make DNA analysis and manipulation possible. These technologies are associated with PCR development of transgenic crops and chromosome painting, as well as DNA sequencing and information processing. The rapid spread of communication technologies enable new knowledge to be disseminated at a remarkable speed worldwide and have ushered in global research initiatives related to crop genome analysis and related research. These tools have been used in various crops for the development of genome maps, QTL analysis, whole genome sequencing, fine resolution mapping, and gene cloning. These scientific advances have also contributed to crop improvement with the application of, for example, marker-assisted selection. The use of molecular techniques has provided a range of new insights into domestication and its future course. Plants have relatively few morphological traits that are controlled by single genes with distinct alleles showing Mendelian segregation. Mendel was fortunate to find 7 independent and non-interacting genes. The development of molecular markers to analyze directly DNA or gene products has meant that tens of thousands of segregating polymorphisms can be analyzed in a species. This has made it possible to locate genes controlling both qualitative and quantitative characters very precisely within the genome, understand their interactions, and then to study the phenotypic effects of individual genes or small regions of chromosomes in segregating populations. In a number of studies, quantitative trait loci affecting different elements of the domestication syndrome are co-located to the same locus or the same small chromosomal region, suggesting either pleiotropy or close linkage of the gene concerned. Fine mapping of genes has led to the ability of clone domestication related genes and unravel the molecular basis of domestication related changes. For example, the 2 genes that are most important in relation to spikelet shattering in rice, that is SH4 and QSH1, have been cloned. SH4 is the key shattering gene that distinguishes cultivated from wild rice, while the QSH1 gene controls the difference in the degree of shattering between some indica and japonica varieties of rice. SH4 is a transcription regulator, and a single amino acid substitution results in reduced shattering. SH4 activates the abscission process, while QSH1 regulates abscission layer formation. Sequence analysis of SH4 has revealed a single base pair mutation that is responsible for non-shattering, and this change is the same in both Indica and Japonica rice varieties. Differences in nucleotide sequence and or levels of transcription of different alleles of transcriptional regulators affect the phenotypes produced by target genes. In wheat, the Q gene is pleiotropic for many domestication traits. The wild type allele, that is small q, is associated with a fragile rachis and grain that does not thrash free of the chaff, whereas the domestication allele q is associated with a tough rachis and free thrashing grain. Comparison of the structure and activity of these two alleles suggests that Q is transcribed at lower levels than Q and that the Q protein functions less efficiently than the protein produced of Q. Q is not known in the wild progenitors of wheat, but human selection post domestication seems to have resulted in upregulation of Q such that Q has more than twice the effect of Q. Similarly, some of the differences in branching and spikelet suppression distinguishing domesticated maize from wild teosinoid and controlled by TB1 have been attributed to upregulation of TB1 in maize. To date, most domestication genes that have been cloned are diverse transcription factors that are usually functional. Thus, the role of human selection on wild populations during crop domestication at the gene level has been modification rather than elimination of gene function. This perhaps reflects the relative rarity of mutations leading to new structural or functional genes and the short time span of crop domestication. Humans caused a major shift in the morphological traits of wild plants by selecting genes of both large effect and small effect to create crops with higher yield of desired product. The wild relatives of crops continue to be an important reservoir of genes for potential use in agriculture. Sometimes the genes that have furnished have had a dramatic effect on yield, and thus there is a continued urgency to conserve these wild genetic resources appropriately, both in situ and ex situ, and to characterize them for future crop improvement. In the major oilseed crop canola or oilseed rap, Brassica jancea, Brassica napus and Brassica rapa, losses of between 10 to 50% of the yield can occur due to unsynchronized pod shattering and require extensive management including spraying with crop desiccants before harvest and windrowing before threshing. Arabidopsis has proved to be a useful model to study the phenomena where a transcriptional regulator, fruitful FUL, mediates pod dehiscence by inhibiting expression of genes controlling shattering. When this transcriptional regulator was introduced in Brassica jancea, it was overexpressed and pods had no shattering. Further, fine-tuning of the expression of this gene in canola may enable the required level of post-harvest shattering to be achieved. Such intentional manipulations to fine-tune gene activity will certainly constitute super-domestication, where genetics interacts with crop management and agronomy. Plant breeding related companies are already looking at the effect of up and down regulating all transcription factors in a given genome aiming to learn more about the target genes of different transcription factors and producing a super domesticate. The rapid reprogramming of biological pathways on polyploidization leads to novel variation that may be exploited by plant breeders. Many breeding programs involve wide and distant hybridization. These procedures cause dramatic genome change, sometimes leading to unpredictable results. Studies of ancient and modern polyploids provide a means of elucidating the effects of dramatic genome change on gene expression and regulation. Results from such studies should enable breeding programs to achieve the desired results. The future course of domestication will continue to rely on changes to the architecture, metabolism, and physiology of crops. To reach new levels of crop yield and new uses for crops, a combination of applied and also theoretical approaches involving computer or systems biology models will be required. To cope with new challenges to crop production, efficient approaches to screen germplasm for genes and both biotic and abiotic stresses in wild and cultivated germplasm are needed. Crops primarily used for food are not also being used to meet demands for sustainable fuel supply, while the use of plants for construction, timbers, and fibers in paper, textile, and composite products continue to increase. To provide value-added crops, new products from crops are being developed. Specialized plant products for processing, such as starches, oils, even plastics for food, pharmaceutical, cosmetic, and industrial uses are required for growing markets. As information accumulates on domestication-related traits and their genome distribution, new avenues to attain higher yield and to tailor-make crops are opened up. Analysis of yield and plant height in a cross between two Japanese rice varieties, Koshikari and Habataki, revealed several QTLs for each trait. One QTL, GN1A, increased grain productivity and acts by altering the production of the enzyme cytokinin oxidase / dehydrogenase that degrades the phytohormone cytokinin. By reducing the expression of GN1A, cytokinin accumulates in inflorescence meristems, resulting in an increased number of grains and hence a plant with the potential for increased yield. By accumulating a variety of yield-related QTLs for increasing both source to produce photosynthate and sink to accept photosynthate, new levels of yield may be achieved. Throughout history, plants have been subjected to changing climate and farmers have adopted new species and varieties to meet the challenges. Indeed, post-glacial climate changes may have been one of the factors leading to the origin of agriculture and plant domestication. Climate change is affecting agriculture in the 21st century. Some changes will be met within existing adaptations of plants, but other factors such as increased UV and carbon dioxide levels require new selections based on understanding of plant responses. A study reported considerable variation in ultraviolet B sensitivity of rice cultivars, which was caused by differences of one or two bases in the CPD, that is cyclobutane pyrimidine dimer photolyase, altering the activity of the enzyme. They suggest that the resistance of rice to UVB radiation can therefore be increased by selective breeding or bioengineering of the genes encoding CPD photolyase. Although carbon dioxide enhancement is regularly used to improve glasshouse production, it is not clear how field crops will respond to changes in atmospheric carbon dioxide concentrations involving complex interactions of photosynthesis with light and dark respiration. There is no naturally occurring waxy wheat variety, but in bread wheat there are waxy loci in each of its three different genomes, that is A, B and D. The waxy locus encodes starch granule protein 1, that is SGP1. Different isoforms are encoded by genes in each genome: SGPA1, SGPB1 and SGPD1. In the germplasm collection of bread wheat, cultivars lacking one of the three isoforms were found. Two cultivars from Korea lacked SGPA1 and one from Japan lacked SPGB1 and one from Turkey lacked SPGD1. By making appropriate crosses, these genes were combined in a single plant, resulting in the first waxy wheat, which had a null for all three isoforms of SGP1, an example of using markers for identification of alleles and then marker assisted selection to find the desired allele combination. The effort to produce waxy wheat in Japan has led to its use as an ingredient for improved Japanese style noodles. These examples show the value of screening germplasm collections with diverse material for useful genetic variation, but also emphasize that it is not always necessary to search in exotic material or employ radical techniques to make innovative progress in plant breeding. We are entering a new era in relation to human understanding of and influence on the genetics of crop domestication. Current research on genes related to crop domestication is providing pieces in the complex jigsaw of gene evolution, networking, regulation and expression in our most important plants. Introduction of alien genes through transgenic technology may be difficult, but continued advances in crop improvement will depend on understanding the genome and its genes. Super domesticates can be constructed with knowledge-led approaches using the range of current technologies. Here we use the term super domestication to refer to the processes that lead to a domesticate with dramatically increased yield that could not be selected in natural environments from naturally occurring variation without recourse to new technologies. The array of genome manipulations that have been developed mainly since the 1980s enable barriers to gene exchange to be overcome and have led to super domesticates with dramatically increased yields, resistance to biotic and abiotic stresses, and with new characters for the marketplace. Hybrid rice can be considered a super domesticate. The teams of scientists that support plant breeders are planning and conducting research to change crops radically. For example, changing crops from C3 photosynthesis to C4 photosynthesis is being proposed because it is now known that plants with C3 photosynthesis have enzymes for C4 photosynthesis, and even well-developed C4 pathways can be found at certain locations in C3 plants. In addition, C4 enzyme genes have been inserted into and successfully expressed in rice. Conversion of a crop from C3 to C4 photosynthesis would certainly be a super domesticate. It was with this background of rapid progress being made in studies of crop domestication that a meeting was organized in Tsukuba, Japan in October 2006 by the National Institute of Agrobiological Sciences and the Organization of Economic Cooperation and Development and supported by Annals of Botany. While the Tsukuba meeting was being planned, a different meeting entitled Plants, People and Evolution, sponsored by the Linnean Society of London, the Systematics Association and Annals of Botany was in preparation. This meeting was held in London in August 2006. Progress in understanding crop domestication and further advances that lead to greater quantities and improved quality of food crops depend increasingly on multidisciplinary team approaches. Various crops have been intensively studied by molecular methods, example maize, barley, tomato, wheat, some of whose genomes have been completely sequenced, example rice and sorghum, or where sequencing projects are proceeding actively, for example soybean and common bean, that is Phaseolus vulgaris, and some where domestication is still at an early stage, example giant cacti, artichoke or banana. The extrapolation information from model species and model crops is already hastening advances in minor crops. This will be particularly true for current genomic initiatives in closely related crops such as the legumes, where data from common bean and soybean will benefit the closely related African and Asian Vigna radiata. Knowledge of the sorghum genome can be tapped to make progress in understanding the complex genome of sugarcane and that of the rice genome for banana. Minor crops, by the very fact that less is known about them, provide the potential of rapidly finding new insights into crop domestication. This is all about today's lecture. Hope you understand well. See you again with a new topic on plant resources. Till then, goodbye.